Hello, welcome to the lecture, and I'm Thomas Ming-Shui Cheng. I've been working in artificial cell for more than 60 years. It's a pleasure to briefly summarize this area for you. First, let's ask the question, does indeed content, cell size, configuration can be varied quite extensively? But the question is, what can you do with all these? This is an example of the possible uses. I'll let you look at it. Microdialysis, drug delivery, blood substitute, biotherapeutic, vaccine, cancer therapy, immunotherapy, enzyme gene therapy, cell therapy, biotechnology, nanotechnology, regenerative medicine, agriculture industry, nanocomputer, nanorobotic, nanosensors. Okay, let's move into the next topic, cancer therapy and immunotherapy. We use artificial cell for the treatment of a skin cancer melanoma. This is based on nano-artificial cell containing nanobiotherapeutic. What we do is that we have hemoglobin and thyroxinase. We're crossing it together to form nanobiotherapeutic. We found that injecting this into melanoma mice normal can decrease the rate of growth but cannot suppress it. So we want to have something that can stay longer at the tumor injected site. So we put these into nano artificial cell so it can sit at the site of action. This is just reported in press, should come out very shortly. We look at 2D culture, we look at 3D culture, we look at injection and the site and how reaction take place. Two D culture result, let's look at this very briefly. We look at proliferation, we look at entry into cell, showing that they enter into the tumor cell and resulting in a decrease in thyroxine level. We also find that they decrease attachment and they also decrease viability of tumor cell, decrease colonization of tumor cell, and decrease metastasis. In 3D culture, please refer to the article that just been published. It shows that thyroxine level is lower, cell count is lower, metastasis is lower, and cell cycle is also starting. We also study in very detail the different components of the artificial cell in affecting the growth of melanoma tumor in mice. You are invited to look at the article, but let's look at a key result. Results shown here. In a control group, giving anti-microcapsule or saline, the tumor continued to grow. But if you give these in nano-artificial cell, it suppresses the growth completely. And the animal body weight doesn't change and there's no side effect. Effect on the growth of high dose of melanoma tumor in mice. This is control. This is showing that with artificial cell, there's inhibition of tumor growth. And the body weight, the growth of animal is unchanged. We notice that occasionally there are some nodule in the nano-artificial cell, so the pathology look at this in more detail, and it shows that In the case of the control, the nodules are all neoplastic, 65% are tumor cell, whereas in the artificial cell one, the nodule, if present, are granuloma, foreign body type of reaction with no tumor cell. Furthermore, there are nanoparticles observed in the nano-artificial cell, none in the control group, and reactions are granuloma reaction. Cancer immunotherapy is a very popular way to also treat cancer. Like Lenker recently published article said that this has become an explosive area, but there are many problems that need to be solved, need for suitable delivery system. I just published an article reviewing the different configuration that you can easily use for cancer immunotherapy. Let's look at this. Let's look at artificial cell and drug delivery, example of some basic principles. There are different basic methods of preparing artificial cell. One of this is by emulsion method to form artificial cell with the membrane. Another approach is nanobiotechnology based on the cross-linking of hemoglobin into polyhemoglobin. Another way is by conjugation, whereby hemoglobin is attached to polymer in the form of polyamine or dextran, or most importantly is polyethylene glycol, PEG. One of the basic methods has been quite frequently used in drug delivery. Drug delivery is based on many approaches. One is polymer crossing protein conjugation. Another one is based on lipid membrane. The first lipid membrane artificial cell is reported by Miller and Rudy. They say they use the 1964 Chang method. Another one is reported by us using lipid polymer complex. Glycogratis from McGill, before he went to Beckham for postdoc, talked to me, and I convinced him to look into artificial cell. So over there, he used Beckham's 1965 lipid onion-like solid microsphere. It's a bit too much lipid and not enough drug can be carried. So as a result, Beckham's group prepared a lipid vesicle called liposome, as based on what they call ether evaporation method. It so happened that we also use ether in our typical method, one of our methods. After this, there is a biodegradable polymer, which is quite useful later. And what is even more exciting is the modification of the surface, polysaccharide, charges, antibody for target delivery. And then very excitingly is PEG by Abusker and Davis group. And this resulted in much improved nano-artificial cell lipid type, polylactic acid type. Now let's look at one interesting thing. It's the COVID vaccine. We always thought that COVID vaccine was developed very quickly, within a year or so when it was required. However, this is not the case. The first proposal of artificial cell for vaccine was back in 1975 when then chair of microbiology at Karolinska invited me to write a chapter on the use of artificial cells for vaccine. This was again published in 1976. Synthetic mRNA was already available by the US group 40 years ago, but nobody saw much interest in either artificial cell or mRNA for vaccine. It was until Pfizer COVID-19 vaccine, more recently based on lipid membrane nano-artificial cell. So instead of starting early to do control clinical trial carefully and to have this produced in large scale, ready for use, because of the delay in interest, they have to rush it, and as a result, very short supply, resulting in many people not being able to receive this when they need this. So this is a good lesson that we should not delay development until it is too late. This was done back in 1976, but at that time, this type of work wasn't that popular yet, and biodegradable at that time, we used polylactic acid that is degraded into lactic acid in the body and become CO2 and water, so it's completely biodegradable. At that time, I prepared nanoparticle or nanocapsule for enzyme or homo vaccine and biology. This is an example using insulin. You can adjust the nanoparticle, capsule, or the microcapsule, microparticle to have different release rate from very slow to very quick or very intermediate. Vaccine, for instance, is one of the ways that was studied. And now you can use it using different polymer, PLA, PEG, PLA, PLGA, and others. You can make microcapsule, microparticle, nanoparticle, polymers, and different names. You can modify the surface to allow to target and other approaches. Another way in drug delivery was initially proposed is to put magnetic material inside artificial cell together with the biology. This way, it's now being used for drug delivery, it's being used in cancer therapy, it's used in laboratory analysis, it's used in bioreactors. Let's look at cancer therapy. One way is to make magnetic nanoparticle containing therapeutic agent or radioisotope, and then put magnetic on the tumor site, and then they would attract the nanoparticle to the tumor site, resulting in localized therapy. And with all of these now, you can combine everything with different configuration of artificial cell, micron dimension, macro dimension for cell, nano dimension, or even nano biotechnological soluble complex of different forms. So as a result, the possible use of artificial cell for cancer therapy and cancer immunotherapy is unlimited and waiting to be thoroughly explored. Finally, may I invite you to visit the website. We don't have time to go into any detail. This is just a very brief summary. If you are interested, the detail in articles, videos, and books are available. If you have any question, I'll be happy to answer, and this is my email address.